Okay, I'm going to talk to you about a DNA structure. Sorry for the slightly abstract title. And some work that really has its origins in pure curiosity, nothing more than that. In the second half, I'm going to tell you about our attempts so far to translate this into doing something that is potentially useful. So I'm going to assume you all know about the DNA double helix. We're in the right place to talk about that. So I'm not going to talk about the double helix. I'm going to talk about a structure called a G-quadruplex, which is actually a four-stranded quadruple helix structure that can form in DNA. Now, the origins of this field go back to the early 1900s when there was an observation that the G base as a monomer at high concentrations could form a gel. This was an observation in a paper published in 1906, but there was no structural data. We didn't even know what DNA looked like at this phase, so it was a recorded observation. In the 60s at the NIH, it's like a game. They showed that what was happening is monomers of the G base at high concentration formed this. And in this structure here, you don't have Watson-Crick base pairing. What you have is, this is the Watson-Crick edge, this is the Hoogsteen edge, and this is a cyclical Hoogsteen-based self-recognition of 4 Gs here with fourfold symmetry. And smack bang in the middle, if you have a positively charged ion of the right characteristics, it stabilizes this array. And of the monovalents, by far, potassium is the very best. And as many of you know, potassium is present at greater than 100 millimolar concentrations in human cells. Now, shown in the middle is a 3D. These are depictions of a crystal structure from Stephen Needle at UCL of the quadruplex formed by the human telomeric repeat system. And you'll see here, beautiful fourfold structure. Now, I started work in this area in 1998 for 2 reasons. I thought this was a beautiful structure and was keen to explore whether it was for real in nature or just a very nice structure in a test tube. And the second reason was esteemed colleagues in the field told me I was wasting my time working on this problem, and why don't I go and do something useful instead? So the combination of the two spurred me on to spend much of the last 25 years on this. Now, in a single-stranded piece of DNA or RNA, the single strand can fold back on itself if it has the right configuration of G bases. And in doing so, in the presence of potassium, it can form a stable 3D structure, and that's the G-quadruplex. I'm going to focus today on this type, this parallel intramolecular quadruplex. Now, to give an overview, some 20-odd years ago, Julian Huppert, who was a PhD student in my group then, when the first draft human genome sequence came out, 2003, he took all the biophysical data that was in the literature and that we generated in our lab and looked at the genome at sequences that had this general motif. And he did a motif search, and he found a couple of interesting things. One, there were lots of motifs in the human genome that we predicted had the propensity to fold into these structures. And 2, the hottest spot in the genome was actually right in front of transcription start sites of protein coding genes. So shown here is a probability density map. This is position with respect to gene structure, transcription start site. This is into the gene. This is in front of the gene. And you'll see here in both strands, blue and red, there's a hot spot about 60 nucleotides in front of the transcription start site. So we and others thought this is an interesting observation, and it led to the hypothesis that maybe these structures can form in one strand in these locations, which are very sensitive locations for the transcription promoting or inhibiting machinery. So this is a regulatory hotspot. Now, over the years, and I should say everything we've done in this area was driven by the need to develop new methods to go to the next stage with the problem. Now, this was what I've shown you as prediction, computational prediction. Now, Vicky and Giovanni developed a method of subverting next-generation sequencing to actually experimentally find these structures in the human genome. And in essence, it works like this. Next-generation sequencing involves immobilizing fragments of genomic DNA on a chip, and you sequence by synthesis of the complementary strand, and you base call by a fluorescent signal at each incorporation. Now, what we do is, first we do this under conditions where lithium is the ion. Now, lithium is not particularly good at stabilizing these structures due to its ionic radius and its hydration energy. So here the polymerase can make the complementary strand and tell us what the sequence is. We then melt away the sequence that's been synthesized, and we reprime and we repeat the experiment, but we replace lithium with potassium, keeping the ionic strength and the pH constant. Now, when we do this, the polymerase encounters an obstacle when these quadruplexes form, and these quadruplexes are very potassium sensitive. And if we compare the first read of the sequence to the second one, we see errors that start to appear when the polymerase encounters an obstacle. So computationally, you can map every site in the genome where this occurs. So we've done this across 12 different species and deposited the data for research use. But in the human genome, again, we found many sites where this could form and saw that the hotspots were in gene regulatory regions, both promoters and also enhancers. Now, it wasn't until it was 2012 when Julia Biffy did these experiments, but we used the Greg Winter method to engineer single-chain antibodies, and here's one which we call BG4, and this antibody recognizes this structural fold in DNA and also in RNA, actually, with 1 nanomolar dissociation constant, and it does not recognize a double helix. So we use this to start looking at cells and tissues, and you can see here in blue is DAPI-stained nuclei of household tissue culture cell lines, and we could see these red punctate stains, which were regions where we believe there are clusters of these quadruplexes forming. And this was really the first time we could sort of see them in cells. And, you know, I enjoyed presenting this data to my friends who told me 10, 15 years earlier that I was wasting my time and why didn't I do something useful. Now I'm going to come back to that. I said to my lab, this is all good and well, but I'm staring at red dots here. How do I know what they really are? And we can't really learn about what they're doing until we can get an address in the genome, in chromatin, of actual human cells. Then we can start to discern what they're doing. Now, Robert Hansel-Hirsch, who runs his own lab now in Cologne, he cracked this problem. Now, over the years, we've generated a number of molecular probes, single-chain antibodies, nanobodies, and synthetic small molecules of different molecular weights. And these probes are all designed, engineered, and validated to bind exquisitely to these structures, but not to canonical B-form DNA. And Robert was the first to carry an experiment of this type. Chromatin immunoprecipitation, which Tony and others have pioneered to study chromatin biology. We can also do with DNA. So here what happens is you can fix chromatin, fragment it, engage with your structure-specific probe, isolate those fragments, sequence them, realign them to the genome, and you see peaks where you accumulate reads, and these peaks correspond to where these structures form. And indeed, they form at sites where the other sequencing methodology I described tells us that they form. But interestingly, most of the ones that can form in naked DNA that you've isolated from cells do not form in the cell. Only about 1% actually form. So chromatin is actually suppressing these structures in cells. But the ones that form in relatively nucleosome-free regions. And you can see here, here's the MYC oncogene, here's a transcription start site, and they're forming in the upstream element. So Robert and others carried out measurements on dynamic changes in whether these structures form. And just because a sequence is there, they don't necessarily form. And what we've shown collectively is that in heterochromatin, where of course there's no transcription, we never see these structures. We've yet to detect them in heterochromatin. Now, in relatively nucleosome-free chromatin, where the gene is transcribing at some low rate, we don't necessarily see them. But when they form, transcription is hyper-elevated. And we also see that these structures form in a way that tracks other canonical epigenetic features, such as histone marks. They are anti-correlated with DNA methylation and chromatin structure itself. So they seem to be a feature in chromatin that is, at the very least, correlated with epigenetics. And if you do sort of lineage tracing of stem cells as you differentiate them, you can monitor the dynamics of the formation and the unformation of these structures at genes as they become activated or inactivated during this transcriptional reprogramming. I just want to throw this in because I'm a chemist. I spend part of my time in the chemistry department and part of my time at the Cancer Research UK Cambridge Institute. Here are a couple of non-drug molecules, probe molecules, that bind to quadruplexes. We developed a technique, this is Zutau and Jochen, for detecting where small molecules bind in chromatin. Two reasons to do this. One, we were interested in these structures, and two, there are so many DNA-targeted drugs that are used every day in oncology and also against pathogens, but we don't actually determine target engagement or look at how they bind, which to me was remarkable. Well, we can do this now. Now, here's an example. We map the binding sites of these molecules in live cells. Here they are. And it's exactly where the antibody detects these structures. And it's this one of the sequencing predicted structures that forms. The other ones, you get no binding and no structure formation. So we can detect small molecule or drug binding to DNA. Now, briefly, and I'm not going to share the data behind this, we've shown that transcription factors, chromatin remodelers, and histone modifiers in the cellular genome seem to be recruited to these structures when they form. And in vitro, we've shown that they bind with low nanomolar dissociation constant to these structures. And this has led to us putting forward a hypothesis that many proteins that appear to be able to bind to the same sequence are actually recognizing structure, not sequence, as their mode of binding. And this is something that Those of you who work in the transcription factor field will know that there's a resistance to new ideas. And so we're enjoying, here we go again, a challenge. But that's our hypothesis. And some evidence for this came from Isabel and Angie, who took the quadruplex in front of the MYC gene, in front of the P1 promoter, used genome editing to take out enough Gs to destroy the structure, and also bring in a different sequence that forms a structure of this type, but from a different sequence. And what they see is, here's the wild type, and we detect quadruplexes with our antibody. When we mutate out Gs, it goes down. And when we swap in a different sequence that can form the same type of structure, we detect it again. This is a control site where nothing changes. And interestingly, when we look at transcription, this is wild type, this is when we mutate out the structure, and this is when we put in a similar structure from a different sequence. So we're seeing structure-dependent control of transcription of this P1 transcript of this very important proto-oncogene, MYC. And some piece of evidence for the protein recognition. Here are two transcription factors, CNBP and SP1, which we and others have shown biophysically combine these structures, and we've shown genomically are recruited to these sites in the genome. Now, when we mutate away the quadruplex, you lose binding of SP1 and CNBP, and when you bring in a different quadruplex, you restore the binding of these transcription factors. So this is part of the mechanism of controlling transcription. Now, Madeline, how long have I got left? Ish. 8 more minutes, okay. I'm going to introduce a company that we formed in 2022 with the name Genome Therapeutics, and stellar team of people associated with this. And it was to build on some of the observations I've described and some observations I'm about to describe. Now here's canonical B-form DNA, and on the left are 3 classes of drugs used to treat most cancers. Every person I know who's had cancer has been treated with one of these 3 classes of drugs at some point. And they are old drugs. Cisplatin, it was 1965 that Rosenberg showed the effect of cisplatin on cells. And it covalently cross-links inter- or intrastrand DNA. Doxorubicin is an intercalator. It slips between the base pairs. Etoposide, people call these topoisomerase inhibitors, and I always say, no, they're not. They interact with an intermediate formed by a covalent interaction of a topoisomerase and a broken strand of DNA that opens up a cavity, and these drugs bind to that complex and trap them long enough to sever the linkage between the protein and the DNA and leave a strand break. Now, these drugs are good and bad. They are cytotoxic chemotherapy. They kill cells that are dividing, which is why if you're treated by them, you suffer quite severe side effects. Yet, 50, 60 years on, we're still using them. They are the most widely used drugs in oncology. So why are we still using them? Anybody? Well, we're using them because they actually work. They are efficacious. Efficacy is not the issue. It's just that it comes at a price. Of course, most of oncology developments have been on protein-targeted therapy, which works very well until you get a mutated amino acid change at the drug binding site. Now, these structures, we and others have shown, they form in a way that's linked to cell states and cell function. And actually, many of the genes where this forms are cancer driver genes. And when they are hyperactive, these structures are very visible for drugging. I will sort of give some evidence about biomarkers as well. But our thinking is these are relatively rare structures, and if you drug them with a small molecule, it's a very different approach to this. So maybe we can generate the kind of efficacy that these drugs generate, but open up a therapeutic window through reduced toxicity. Now, here's something I haven't shared. These structures are much more abundant in cancer cells than non-cancer cells. This is one example of a cancer normal paired biopsy, and the dark stain is that antibody probe lighting up the structures. You see the same in cell lines. This is a typical breast cancer model cell line. This is a normal breast epithelial cell line. Even by eye, you'll see that there are far fewer spots. We've also mapped these out genomically, and we see generally cancer states have far more of these structures than non-cancer states. Now, we can drug these structures with molecules that don't interact with bDNA. That's great. There are more of them in cancer states, more targets. That's also good. Now, some work we did a while back with Steve Jackson, who many of you will recognize, and Sam Aparicio, that some of you will know, showed that by binding drug-like molecules here, we could cause strand breaks at these sites. So they're site-specific strand breaks that depend on the formation of these structures. And the drug binds, and then the actual cleavage mechanism we're still working the details of. It has some dependence on replication and some dependence on transcription. And you can detect this by fairly standard assays for the downstream effects of strand breaks. So this was an interesting observation. And here's the premise of what's going on in genome therapeutics. In healthy normal cells, there are far fewer of these structures. If you happen to bind them with a drug, and that leads to a productive strand break, it actually gets repaired very efficiently. In cancer cells, there are far more targets, far more target engagements with the drug. And we've shown by extensive functional genomic analysis an array of genetic backgrounds where this effect is severe. And they are all shared by, if I can group cancers into a bucket called genomically unstable cancers, those cancers, you get high ligand binding efficiency because of the abundance of the target, and you only need one unrepaired double strand break to cause lethality. So this is synthetic lethality. Now, the company has advanced preclinical compounds, and here's just a sample of data. This is a cell model data. This is HCT116, where we've taken out the BRCA2 gene, both alleles, and MCF10A, and this is one of our advanced compounds. And this is the therapeutic window. This is the cell killing of cells with that genetic background versus wild type. So 3 orders of magnitude difference. Now, in the same experiment, if you try cisplatin or etoposide or doxorubicin, these cytotoxic chemotherapy agents, the therapeutic window here is barely 2-fold or 3-fold as compared to 3 orders of magnitude. So this seems to support the thinking from the science. This is a few months old now. This is an animal model study, control versus the same drug candidate here. You see it melting away the tumor. Here we're only dosing once a week by IV. And what's not shown here is 3 weeks after the last dose, the effect has sustained. So there's a long-term durability to this. So where we are is now pushing this towards a clinic. We're hoping to be in the clinic back end of next year. And this all started really with curiosity and a bit of inspiration from a naysayer. So there are many people in my lab over many years, we've been doing this for 25 years now, who've contributed to where we've got to. I only gave a snippet today. These collaborators, and more recently the company, Genome Therapeutics. This is how I'm funded. Most of the fundamental work was supported by Cancer Research UK. Thank you.
Oh, wow. I see a lot of questions. Somebody's got a microphone over there, so let's start with you.
Is this working?
Yeah.
Hi, Christian Dillon, CSO at Foremost. Hi.
Hi.
Fantastic talk, thank you. Yeah, two-part if I might be permitted. So first question, the G-quadruplexes, presumably they need to be resolved for effective DNA replication to occur. So do we know anything about that process? And second question...
Sorry, could you repeat that first question?
Yeah, the first question was for effective DNA replication. Presumably the quadruplexes need to be resolved. Do we know anything about that?
Yep.
And then the second question, you mentioned you get quadruplexes on RNA. Is that mRNA or different RNA species? And do we know anything about the function of quadruplexes on RNA?
Yep. So first question, absolutely right. These structures need to be resolved, the replication fork. And there are particular helicases that seem to have been evolved to do the heavy-duty unwinding of these knots during replication. And we helped solve the X-ray structure of one of them called DHX36, bound to a quadruplex, and it reveals the mechanics of how these helicases recognize and then resolve these structures. And interestingly, I mean, there are other helicases, Blooms and Werners as well. Now, when these specific helicases are dysfunctional, that is actually one of the genetic backgrounds that leads to genomically unstable cancers. So there's a linkage there. RNA does occur in RNA. Now, we've done many experiments. There's a whole field there of what does it do. The effects in RNA do manifest in messenger RNA. And two areas in messenger RNA where there's clear evidence. One is in 5' UTRs, they can interfere with ribosome assembly and processing. And in fact, they also occur in the LTRs of RNA viruses, retroviruses, such as HIV-1 and SARS-CoV-2. The other place, which is a hot spot, is at the first intron-exon junction. And there's evidence, again, from genome editing that if you manipulate the quadruplexes at these intron-exon junctions, that you can influence splicing. We've done some work, some of it with Tony, on pre-microRNA and microRNA processing. So there appear to be some pre-microRNAs where these structures can form and they can influence the processing, the maturation process of microRNAs. But the third area I'll highlight is stress granule formation, which is quite topical in the condensates field. And there's some evidence of a linkage of certain repeat expansion units that can form RNA quadruplexes and sequester proteins as part of formation of stress granules.
Tony has a question.
Tony.
Can you tell us a little bit about what you know about the recognition selectivity of G-quadruplexes for transcription factors? So do you think they bind directly, or would there be proteins that bind specifically to G-quadruplexes and therefore recruit by protein-protein interaction transcription factors?
I think both are possible, but to give one example, SP1 I didn't talk much about it, but we've really taken apart, and SP1 binds directly. The DNA binding domain only of SP1, which is a 3' zinc finger protein, it binds with low single-digit nanomolar KD. So there are transcription factors that seem to bind it directly.
Good. Okay, I think, let's squeeze in one more question. Let's have this be the last question.
You hinted at the concept of proteins binding to these, and is it possible that some of the other ones you predicted from your naked DNA aren't being made, but are just masked by reader proteins?
It's possible, because if the protein binding prevents our probe from binding, we wouldn't see it. So if it's a static 100% coverage scenario, we would miss it. So that is a possibility. But usually with regulatory proteins that interact with DNA, temporally they're hopping on and off and across the cell population. It's usually not 100%. So I think it's theoretically possible, but probably unlikely that we've completely missed it. Good question.
Okay, let's thank Shankar one more time.